Welcome dear students. Today we will be discussing about secondary metabolites. The main objectives of today's discussion are to understand the role of secondary metabolites in plants, to study the types of plant-derived secondary metabolites, to comprehend the beneficial effects of plant phenolics on humans, to realize the bioavailability of polyphenols, to read about the secondary metabolites from microbes. The chemical reactions that take place in any organism is called metabolism. The food that the organisms contain in carbon, nitrogen and mineral gets converted into energy. The energy produced ends up in molecules that are common to all cells. These molecules are required for the proper functioning of cells and organisms. For example, lipids, proteins, nucleic acids and carbohydrates, etc. They are also called primary metabolites or biomolecules. The primary metabolites are involved in photosynthesis, respiration, protein and lipid metabolism. Most plants also spend carbon and energy for the synthesis of molecules that do not seem to have any role in normal cell function. These molecules are known as secondary metabolites. Now dear students, let's learn about the various roles of secondary metabolites. They induce plant defense against insect infestation. They help to recognize specific components of insect saliva. Some of these compounds inhibit herbivore digestion. Herbivore-induced volatiles have complex ecological functions. They behave as antimicrobial compounds against pathogen attack. Infection induces additional antipathogen defense. Phytoalexins increase after pathogen attack. Some plants recognize specific pathogen-derived substances. A single encounter with a pathogen may increase resistance to future attacks. Now let's discuss types of plant-derived secondary metabolites. Plant secondary metabolites can be divided into three chemically distinct groups: terpenes, phenolics and nitrogen containing compounds. First we will discuss terpenes. The terpenes or terpenoids are the largest group of secondary metabolites produced by plants. Most of the compounds in this class are insoluble in water. Terpenes may or may not have well-defined functions in the plants. For example, gibberellin, a diterpene, is an important plant hormone and is a primary metabolite. The vast majority of terpenes, however, are secondary metabolites presumed to be involved in plant defense. Terpenes deter the feeding of herbivores and are toxins to many herbivorous insects and mammals. Thus, they play as defense molecules in the plant kingdom. For example, pyrethroids, which are monoterpene esters found in leaves and flowers of chrysanthemum species, have insecticidal activity. Monoterpenes accumulation in resin ducts of conifers such as pine and fir are abundant in the needles, twigs and trunk. These compounds are toxic to bark beetles, which are serious pests of conifer species throughout the world, and to numerous other insects. Essential oil is a mixture of volatile compounds commonly found in plants, and they lend a characteristic odor to their foliage. Essential oils have well-known insect repellent properties. For example, peppermint, lemon, basil, that's tulsi, and lemongrass. Monoterpene of lemon oil is called limonene and that of peppermint oil is called menthol. The presence of both lipid soluble, that is the steroid or triterpene, and water soluble, that is the sugar elements, in one molecule gives saponins detergent like properties. Both natural and synthetic pyrethroids are popular ingredients in commercial insecticides because of their low persistence in the environment and their negligible toxicity to mammals. Secondly, we will discuss phenolic compounds. Most of the plants produce a large variety of secondary compounds that contain a polyphenol group. These substances are classified as phenolic compounds or phenolics. Plant phenolics are chemically heterogeneous group of nearly 10,000 individual compounds, some of which are soluble only in organic solvents. Some are water soluble like carboxylic acids and glycosides, and others are large and insoluble polymers. Phenolics play a variety of roles in the plant due to their chemical diversity. Many serve as defense against herbivores and pathogens. Others function in mechanical support in attracting pollinators and fruit dispersers, in absorbing harmful ultraviolet radiation, or in reducing the growth of nearby competing plants. One of the major group of phenolic compounds are tannins. Tannins are astringents, bitter plant polyphenols that either bind and precipitate or shrink proteins. These tannins are usually divided into hydrolyzable tannins and condensate tannins called proanthocyanidins. Tannins, the most abundant and widely distributed phenolic polymers of higher plants, combine with proteins, polysaccharides, etc. to form complexes, thereby increasing their resistance to microbial degradation. As already said that there are two major groups of tannins, that is hydrolyzable and condensate tannins. Hydrolyzable tannins are composed of esters of gallic acid called gallotannins or ellagic acid called ellagitannins with a sugar core which is usually glucose. These are readily hydrolyzed by acids or enzymes into monomeric products. The major commercial hydrolyzable tannins are extracted from Chinese gall, that is Rhus simulata, sumac, that is Rhus coriaera, Turkish gall, that is Quercus infectoria, tara, that is Caesalpina spinosa, myrobalan nuts, that is Terminalia cibula, and chestnut, that is Castanea sativa. Condensate tannins, also known as polymeric proanthocyanidins, are composed of flavonoid units and are usually more abundant in tree barks and woods than their hydrolyzable counterparts. The important commercial condensate tannins are extracted from wattle, that is Acacia mollissima and Acacia mansiae, Cubraco, that is Chinopsis lorenzi and Chinopsis balancea, and tree barks. The colored pigments of plants help to attract pollinators and seed dispersers. The colored pigments are of two types, that is carotenoids and flavonoids. Carotenoids are yellow, orange and red terpenoid compounds. Carotenoids also serve as accessory pigments in photosynthesis. The flavonoids include a wide range of colored compounds. The most widespread group of pigmented flavonoids is the anthocyanins. Anthocyanins are responsible for most of the red, pink, purple and blue colors seen in flowers and fruits. The other two groups of flavonoids like flavones and flavanols are commonly found in flowers. These flavonoids absorb light at shorter wavelengths than anthocyanins. Hence, they are not visible to the human eye. However, insects such as bees, which can see the ultraviolet range of the spectrum, respond to flavones and flavanols. Isoflavonoids, which are found mostly in legumes, have several different biological activities. Some, such as rotenone, can be used effectively as insecticide, pesticide, for example, as rat poison, and pesticide, that is fish poisons. Other isoflavones have anti-estrogenic effects. For example, sheep grazing on clover rich in isoflavonoids often suffer from infertility. Isoflavones may also be responsible for the anti-cancer benefits of foods prepared from soybeans. A second category of plant phenolic polymers with defensive properties besides lignin is the tannins. They are general toxins that can reduce the growth and survival of many herbivores when added to their diets. In addition, tannins act as feeding repellents to a great variety of animals. Mammals such as cattle, deer and apes characteristically avoid plants or parts of plants with high tannin contents. Unripe fruits, for instance, frequently have very high tannin levels, which deter feeding on the fruits until their seeds are mature enough for dispersal. Herbivores that habitually feed on tannin-rich plant material appear to possess some interesting adaptations to remove tannins from their digestive systems. Plant tannins also serve as defense against microorganisms. From leaves, roots and decaying litter, plants release a variety of primary and secondary metabolites into the environment. The release of secondary compounds by one plant which has effect on neighboring plant is referred to as allelopathy. If a plant can reduce the growth of nearby plants by releasing chemicals into the soil, it may increase its access to light, water and nutrients. Allelopathy is currently of great interest because of its potential agricultural applications. The reduction in crop yields caused by weeds or residues from the previous crop may in some cases be a result of allelopathy. An exciting future prospect is the development of crop plants genetically engineered to be allelopathic to weeds. Now let us know about some beneficial effects of plant phenolics on humans. These include anticoagulant activity akin to aspirin, preventive action against cancer, combating the development of tumors, powerful antioxidant activity which is 20 times more than vitamin E, anti-inflammatory effects and facilitating the reaction of the immune system. However, all these effects are under the administration of small quantities of these phenolic compounds. Higher quantities are poisonous. An important part of this discussion is the bioavailability of polyphenols. It's important to realize that the polyphenols that are most common in human diet are not necessarily the most active within the body, either because they have a lower intrinsic activity or because they are poorly absorbed from the intestine, highly metabolized or rapidly eliminated. In addition, the metabolites that are found in blood and target organs and that result from the digestive or hepatic activity may differ from the native substances in terms of biological activity. Extensive knowledge of the bioavailability of polyphenols is thus essential if their health effects are to be understood. Metabolism of polyphenols occurs via a common pathway. The glycones can be absorbed from the small intestine. However, most polyphenols are present in food in the form of esters, glycosides or polymers that cannot be absorbed in their native form. These substances must be hydrolyzed by intestinal enzymes or by the colonic microflora before they can be absorbed. When the flora is involved, the efficiency of absorption is often reduced because the flora also degrades the glycones. During the course of absorption, polyphenols are conjugated in the small intestine and later on in the liver. This process mainly includes methylation, sulfation and glucuronidation. This is a metabolic detoxification process common to many xenobiotics that restricts their potential toxic effects and facilitates their biliary and urinary elimination by increasing their hydrophilicity. The conjugation mechanisms are highly efficient and aglycones are either absent in blood or present in low concentrations after consumption of nutritional doses. Circulating polyphenols are conjugated derivatives that are extensively bound to albumin. Polyphenols are able to penetrate tissues, particularly those in which they are metabolized, but their ability to accumulate within specific target tissues needs to be further investigated. Polyphenols and their derivatives are eliminated chiefly in urine and bile. Polyphenols are secreted via the biliary route into the duodenum, where they are subjected to the action of bacterial enzymes, especially beta-glucuronidases, in the distal segments of the intestine, after which they may be reabsorbed. This enterohepatic recycling may lead to a longer presence of polyphenols within the body. The third type of plant-derived secondary metabolites, as mentioned earlier, are nitrogen-containing compounds. These compounds are commonly known as alkaloids. In next part of today's deliberation, we will discuss the secondary metabolites from microbes. In the microbial system, the phase of growth is well defined. Primary metabolites are involved in growth, development and reproduction of the organism. The primary metabolite is typically a key component in maintaining normal physiological processes. Thus, it is often referred to as a central metabolite. Primary metabolites in microbes are typically formed during the growth phase as a result of energy metabolism and are considered essential for proper growth. Examples of primary metabolites include alcohols such as ethanol, lactic acid and certain amino acids. Secondary metabolites are typically organic compounds produced through the modification of primary metabolite synthesis. Secondary metabolites do not play a role in growth, development and reproduction like primary metabolites. They are typically formed during the end or near the stationary phase of growth. Many of the identified secondary metabolites have a role in ecological function, including defense mechanisms, by serving as antibiotics and by producing pigments. Examples of secondary metabolites with importance in industrial microbiology include atropine and antibiotics such as erythromycin and bacitracin. Secondary metabolites in the microbial world serve as competitive weapons used against other bacteria, fungi, amoebae, plants, insects and large animals, as metal transporting agents, as agents of symbiosis between microbes and plants, nematodes, insects and higher animals, as sexual hormones and as differentiation effectors. Secondary metabolites are not produced until the microbe has largely completed its logarithmic growth phase and entered the stationary phase of the growth cycle. Period of production is called as stationary phase. It is also known as idiophase and metabolites are known as idiolytes. In the idiophase, cells do not divide but are metabolically active. Idiolytes are organic compounds produced only after considerable number of cells and primary metabolites have accumulated, that is, at the end or near the stationary phase of growth. These are produced under suboptimal concentrations of oxygen, deviations of pH or when primary nutrient source is depleted. Though idiolytes are a characteristic feature of fungi, yeast, actinomycetes and bacterial growth, but these are not produced by a few strains of Escherichia coli. In some strains, secondary metabolites are produced by further conversion of a primary metabolite. Alternatively, these may be metabolic products of the original growth medium, not necessary for growth, development and reproduction like primary metabolites. Their production is influenced by environmental factors. Secondary metabolites are synthesized for a definite period by cells that are no longer undergoing balanced growth. A single microbial type can produce very different metabolites. Their production is regulated by complex biochemical pathways, and some strains can produce a variety of ideolites. For example, a strain of Streptomyces can produce a variety of 35 anthracyclines. Examples of secondary metabolites from the microbial world are antibiotics, toxins and pigments, to name a few. Bacitracin, an antibiotic peptide derived from Bacillus subtilis, is commonly used as a topical drug. It has a wide spectrum antibiotic activity against gram-positive and gram-negative bacteria. Common toxins from the bacterial world are exotoxins. Some examples are Corynebacterium diphtheriae. Exotoxin inhibits protein synthesis and causes cell death, causes diphtheria. Clostridium tetani. Exotoxin blocks action of neurons on spinal cord, causes tetanus. Clostridium perfringens causes gas gangrene. Exotoxin is called alpha toxin, which causes cell death. Enterotoxin causes hypersecretion of water and electrolytes in diarrhea. Clostridium botulinum. Exotoxin causes paralysis of respiratory muscles by attacking acetylcholine of synapses and neuromuscular junctions, leading to death, causes botulism. Vibrio cholerae strain 01 and 0139. Enterotoxin causes hypersecretion of water and electrolytes within gut in diarrhea. Enterotoxigenic Escherichia coli produces enterotoxin, which is a heat labile exotoxin, and causes hypersecretion of water and electrolytes within the gut. Shigella dysenteriae type 1, that is Shiga bacillus, exotoxin causes acute inflammation. Staphylococcus aureus secretes toxic shock syndrome toxin 1, which causes toxic shock syndrome. Staphylococcal enterotoxin causes toxin type food poisoning and stimulates vomiting center of brain. Streptococcus pyogenes secretes pyrogenic or erythrogenic exotoxin, which causes scarlet fever and toxic shock syndrome. In addition, pigments from microbes are being used as a replacement for artificial colors in food, pharma and healthcare industries. Overproduction of secondary metabolites can be achieved by manipulating larger number of genes called cassettes. Now dear students, let's conclude here. Nature produces an amazing variety and number of products. About 1 lakh secondary metabolites with molecular weight less than 2500 have been characterized, mainly produced by microbes and plants. Some 50,000 are from microorganisms. New bioactive products from microbes continue to be discovered at an amazing pace, that is 200 to 300 per year. Many companies have participated in the successful discovery and application of microbial secondary metabolites. This is all about today's deliberation. I hope you have understood well. Thank you very much.